Depends on how quickly we move. All right. So we have, I think, actually one more question to deal with regards to the light-dependent reactions. And then we'll take ourselves a lecture break and move into the light-independent reactions of photosynthesis and talk about some different mechanisms or some different strategies by which you carry out those light-independent sections. All right, so the last question for the light-dependent reactions. How do electrons flow through the photosystems? We've already talked about this a little bit. So chlorophyll A is capable of just shuttling energy. We left off actually on Wednesday with the diagram that when an electron gets excited, it can drop down and emit light to a neighboring pigment molecule, or it can transfer to a neighboring pigment molecule, or it can transfer to an electron acceptor in the reaction center, right? You have three options for those electrons. So these chlorophyll A molecules, the primary pigment in plants and green algae, pass electrons to primary acceptors in two photosystems. So there's photosystem 1 and there's photosystem 2, both of which have certain features. Both of which have what's called an antenna complex or an antenna complex. And this is where you have a whole collection of chlorophyll pigments surrounding a single reaction center. And the reaction center is a specialized chlorophyll A. And so usually when you see a cartoon representation of this, it's nothing like what it actually looks like, but it visually helps you to understand a little bit better as electrons are moving, which is nice, right? Sometimes models are not entirely accurate, but are helpful because they're not entirely accurate. So each photosystem has, an antenna complex, a whole collection of chlorophyll pigments, and then a specialized chlorophyll pigment in the middle called the reaction center. Photosystem 1 has a reaction center that is triggered or optimized at 700 nanometers towards the top of the visible spectrum. What color light is this? Anyone know? Red. This is red light. So photosystem 1 has a reaction center that works most optimally at 700 nanometers. Photosystem 2 has a reaction center that works optimally at 680 nanometers. What color light is this? Still red. Getting closer to orange though. Getting a little bit more high energy, but both of these lights would actually be red. This is higher energy light than this, but not incredibly different in terms of the amount. Why is this one higher energy than this? Yeah, so wavelength and energy are inversely proportional. As wavelength decreases, energy increases. Yeah.
So if you're growing plants and you have a red light over them, all 24/7, that would help them grow more.
Potentially, although they'll absorb other wavelengths of light, and then they just bounce it around until it gets to the right wavelength in the reaction center. So they'll absorb other colors of light. Mm-hmm. But yeah, if you gave them only red light, they'd probably perform a little bit better than they would with just white light, as long as the intensity was the same. Now, if you use like a red filter, so it filtered out some of the intensity, and then you're getting red light, it's not going to perform as well. But if you gave it the same intensity light, the same number of photons, and they were only red, it would probably perform better. Okay. Yeah. All right. So all of the pigment molecules in the antenna complex absorb light. However, they are not going to they're not going to take electrons and pass them to an electron acceptor and get the electron transport system started. They may pass electrons, but most often they're just going to pass light. Their electrons get excited, drop back down, emit light that's absorbed by a neighboring pigment molecule. And this whole process is called inductive resonance. And I made this analogy last time. It's an okay one. I'm not going to say it's a great one. It's an okay one, like a game of hot potato, right? You're not quite ready, so you pass it to the next person. The analogy isn't perfect because it really doesn't accurately portray what happens at the reaction center. All right, so again, here's the representation of this photosystem. It's not a perfect image of what the photosystem actually looks like, but it's actually a really good representation of what's going on. And so here you have the antenna complex packed full of chlorophyll A. Here you have the reaction center, which has a specialized chlorophyll A that is capable of stripping electrons and sending them to a really high energy level to the primary electron acceptor. Now, where do the electrons come from? From water. And what is it that sends it up to this really high energy level?
The light.
The light. And what frequency of light if this is photosystem 2? Or what wavelength? Frequency, you'd have to do some math. 680 nanometers. If it's photosystem 1, what wavelength? 700. Both cases still dealing with red light. And so it's only at that proper wavelength of light that you can strip the electrons away from water and send them up to a higher energy level. Now in photosystem 1, the electrons are not coming from water. So keep that in mind. In photosystem 2, they are. In photosystem 1, they're coming from the electron transport system.
Why can't they use a higher frequency? Like if they have really strong light, wouldn't that bounce it up too?
Yes, but not to the precise. So you have to the primary electron acceptor has a very specific energy. It's an orbital, right? Electrons can only exist in orbitals, and every orbital corresponds with a very specific amount of energy. So if you have too much energy, it would send it up too high, and it would not sit in that orbital. It would go up higher, which would mean it would need to bounce back down to a lower energy level.
What's wrong with it being too high?
Well, then it's just too high energy to be in that orbital. That's why it got bounced out of the orbital in the first place.
But if it's like in a higher orbital, what's the problem?
Because it's a higher orbital than what the acceptor has available.
Okay.
We can talk about it
Okay.
another time. All right, so keep working through this. Remember, the electrons for photosystem 2 are coming from water. And again, they're going to get excited in this reaction center where you've got the specialized chlorophyll A. And in photosystem 2, it's going to take light with a wavelength of 680 nanometers, use that light to strip the electrons away from water and send them to a primary electron acceptor. And then once they get here, the electrons are going to do what electrons do best, and that's find a lower energy orbital to be in. And as they do, they bounce from one electron acceptor to another through an electron transport system very similar to the one that we saw in cellular respiration, okay, where the electrons move from one piece to another, and as they do, they drop in energy, and as they drop in energy, you can use that energy to fuel other processes. Now, where it's different, you have different electron carriers. In the electron transport system in cellular respiration, what are our electron acceptors? In cellular respiration. Well, so it starts with NADH. So this is cellular respiration. It's not photosynthesis. So you've got NADH and FADH2 that were reduced in steps 1, 1A, and 2, right? But your electron acceptors, the first one is either complex 1 or complex 2, depending on whether the electrons came from NADH or FADH2, right? So that's one of your electron acceptors. Actually, that's two of them, complex 1 and complex 2. And then complex 3 and complex 4, right? And the final electron acceptor in cellular respiration is oxygen. And then you also have cytochrome C and ubiquinone as additional electron acceptors. So the electron acceptors are different in photosynthesis, but the overall process is the same. Instead of having complex 1, complex 2, complex 3, complex 4, the cytochrome C and ubiquinone, like you have in cellular respiration, you have what's called plastoquinone, which is very similar to ubiquinone, which is why it has a very similar name. But plasto, what does that root mean? It means plant. And then plastocyanin, another electron carrier. And so as electrons drop, they release free energy. And like in our electron transport system in cellular respiration, the cells use that energy to pump hydrogen ions against their concentration gradient. And again, unlike cellular respiration, oxygen is not the final electron acceptor. Oxygen was made during this process, but that oxygen diffuses out of the plant's tissues. Instead, NADP plus is the final electron acceptor. NADP plus is the final electron acceptor. And the enzyme that catalyzes the transfer of electrons to NADP plus is called NADP plus reductase or NADP plus dehydrogenase. Two names for the same enzyme. Okay, I'll show you a picture of this. It'll start to put it all together. We'll have a lecture break that will hopefully continue to put it all together. All right. So photosystem 1 can work independently of photosystem 2. Now photosystem 2 comes first. Photosystem 2 strips the electrons away from water, transfers it down the electron transport system, which photosystem 1 is part of. But photosystem 1 can work without photosystem 2. We call this cyclical electron flow when photosystem 1 is operating independently. So instead of electrons being passed from photosystem 1 to the final electron carrier, which is what? Or the final electron acceptor. Now, it's oxygen in cellular respiration. It is not oxygen in photosynthesis, NADP+. So instead of electrons going from photosystem 1 to NADP+, they go to a molecule called ferredoxin and end up back in the reaction center of photosystem 1, where they can be excited again. By what? What's exciting the electrons in photosynthesis? Light, right? That's what the root photo means. And so as it does this, it continues to make ATP because hydrogen ions are continuously pumped against their concentration gradient as those electrons flow, but the electrons are never passed to NADP+, right? They just end up coming back to photosystem 1 and get excited, go to ferredoxin, come back to photosystem 1. Get excited, go to ferredoxin, come back to photosystem 1. And so that whole time you're shuttling hydrogen ions against their concentration gradient, facilitating making ATP, but you do not generate any NADPH. All right? Okay, we're going to take our lecture break. Let me show you some pictures first. So here's what's called linear electron flow. In linear electron flow, you use both photosystems. Where do the electrons come from initially? From water. They get excited by light that's at 680 nanometers. They pass from this acceptor in the reaction center to plastoquinone, to plastocyanin, to photosystem 1, where they get excited again by light at 700 nanometers. And then from there, they pass over to NADP+, the final electron acceptor. And notice these electrons are at a much higher energy level on NADPH than they were at the water. Okay, so you have electrons fairly unstable, which means NADPH is going to be really good at doing what?
Oxidizing?
Yeah, at oxidizing, at losing those electrons. Which is also true of NADH, and also true of FADH2, which is why they're wonderful electron carriers, because they don't hold on to those electrons really tightly. If they did, that's not a good carrier, right? A carrier should transport things, not keep them. That's a hoarder. It's not a transporter, right? Make sense? We okay? So this is called linear electron flow, as opposed to cyclical. There's just all the machinery inside of the membrane. There you can see the hydrogen ions that have been pumped against their concentration gradient into the lumen of the thylakoid, now flow through ATP synthase, generating ATP. And here's cyclic electron flow. Notice we're only using photosystem 1. The electrons get excited there by light at 700 nanometers. Instead of passing to NADP+, they pass over to ferredoxin and then back into photosystem 1. Okay, and get excited again and flow to ferredoxin and then back into photosystem 1, generating a great deal of ATP, but not generating any NADPH. Okay, it's time for a lecture break. What I want you to do, working in your groups, which I know some of the the quiz groups disassembled as people migrated back to where they're comfortable. But what I want you to do in your groups, hopefully of 5 or 6 individuals, is come up with what would cause a cell to shift from linear to cyclic. Why would a why would a cell make that shift from linear to cyclic? And what's the value of making additional ATP if you're not actually reducing NADP+? So why is there value in photosystem 1? And what would cause a cell to shift over into just doing the cyclic? All right? Take a couple of minutes, talk about it with those around you, see if you can come up with a list of advantages of just doing the cyclic and a list of conditions that would cause the cell to shift over to it. All right? 2 minutes, starting now.
But I don't know how the dark reacts you need an NAD you need to reduce that ADP thing to make glucose, right? How can you make glucose?
Well, if you don't have the calories.
Like, can you make glucose?
So like in mediation,
So the ATP cannot be used to just making an energy, but it does. Oh, I bet it's when the cell itself just uses it like
So, yes.
It's not longer.
You don't have to come up with new electrons. It's in the same electron. So
yeah, and
the electrons are coming from water.
What do you have with water?
The cell's dehydrated. So you know how the light comes
Oh, I do know how the light comes in and bounces.
So it's when it's dehydrated and then you use energy right then for that. It's not for the. Because it can't give the ATP, like it can't transfer ATP.
Oh, increase the weight. Yes.
Because that's right.
So it's going to be absorbed, and then as the electron falls back down, it's going to be
Right, but so that's the cell itself that needs it.
right. Certain tissues.
and even higher energy, like
So I guess like
higher energy,
it's when like the little cells producing glucose is like
get down to 600.
yeah. It's like it's not helping for anybody else.
All right, take another, 10, 15 seconds, wrap up your thoughts. Find somebody to represent the thoughts of your group.
Do you need NADPH
To produce glucose? Yes.
So you can't do it that way. Sure.
Represent.
Caleb.
All right. So, let's start with the question: what is the advantage of cyclic electron flow? So, what's the advantage of it? So we have Kaylin. Do we have any other takers? Okay, Kaylin.
Okay, our group talked about, it was actually Joey's concept, talking about how when you shift from linear to cyclical, and the cyclical, you're reusing electrons, and you don't have to constantly have you do need some energy, but it's not a continual inflow of energy, it's more of a.
Well, yeah, you need energy in the form of sunlight,
Right.
but you don't need any new electrons. You just keep reusing the same electrons, which is fantastic, right? You're not recycling the electrons because they're not being used to do anything different. You're reusing them, right? So it allows you to continually reuse the same electrons. It absolutely is an advantage of the cyclic electron flow. Any other advantages? Yeah, Jonathan.
Less water.
Requires less water. It's good. Yeah. Any other advantages? You said the same things. Another advantage is it just generates a lot more ATP, right? So NADPH we haven't really talked in any way about what a cell uses that for, other than that it uses it in the light-independent reactions. But you know what a cell uses ATP for. Everything, right? So sometimes the cells just need to generate a lot more ATP without actually using the sugars that they made in the process of photosynthesis. All right. Now, under what conditions would a cell shift over into cyclic electron flow? Yeah.
Maybe when there's not enough water?
Yeah, when water content is too low, so there's no source for new electrons, right? There are lots of times when water flow is too low. Think about plants that are drought tolerant, that live in places where the soil dries up quite a bit. All right. Any other ideas under other conditions that might shift a cell into cyclic electron flow? If you get a buildup of NADPH, right? We've talked about this before. Oftentimes, the products of an entire process act as a negative feedback on the process. And NADPH, having excess NADPH, can actually shut down the work of this enzyme here. NADP plus reductase or NADP plus dehydrogenase, whatever you want to call it. Okay? So that's another condition that could shift the cell over into cyclic electron flow. And another condition is when ATP levels are really low. When ATP levels are really low, it can shift the cell over into cyclic electron flow. All right. Any questions about the light-dependent reactions of photosynthesis? Yeah.
How is one not dependent on, how is two independent but one isn't? And one's the first one? Like,
Well, one is called one because it was the first one discovered.
Right.
One isn't called one because it's, I guess it's also, it can function independently of two, but two cannot function independently of one.
How can two not function independently if it's the first one? Like, wouldn't it just start with two, but then not finish all the way?
No, because there's no way, once two excites those electrons, there's no way to get those electrons back to two. You have to have a new source of electrons. You have to constantly have a source of new electrons to run photosystem 2. Because there's no way to get those electrons back there. You don't have to have a constant source of new electrons to run photosystem 1. All right? So our last 4 important questions dealing with the light-independent reactions of photosynthesis. First one, what are the basic processes? Second one, how does the Calvin cycle produce sugars and other molecules? And then the last 2, which your text did not talk about, your quiz did, and we shall. And then we'll finish up and y'all can, frolic off into spring break. Unless, of course, you have a class after this and you can frolic off to that class or stay in the same room if your next class is in here. All right. So what are the basic processes of the light-independent reactions? All right. During the light-independent reactions, we call this the Calvin cycle. Electrons pass from NADPH using energy from ATP, and we fix carbon. We reduce carbon. Okay, so that's big picture what's going on during the light-independent reactions. During the light-dependent reactions, what did we reduce? NADP plus. But we know that in photosynthesis, carbon has to get reduced, so that way you have the availability to do cellular respiration where carbon gets oxidized, right? Okay, so the Calvin cycle, the light-independent reactions, have 3 phases. The Calvin cycle has 3 phases. The first one is called carbon fixation. And this is where you take carbon dioxide and you attach it to another molecule. In this case, you attach it to RUBP, which stands for ribulose 1,5-bisphosphate. And so as far as what I expect you, if you are comfortable with RUBP, I'm fine with that. You don't need to know ribulose 1,5-bisphosphate. If you want to, that's fine as well. I'm fine with that. All right, and the enzyme that facilitates that reaction is called rubisco. Again, if you want to know that that's ribulose 1,5-bisphosphate carboxylase oxygenase, I'm fine with that. But if you know it as rubisco, that's cool. All right, rubisco is not found in any organism that is not photosynthetic. Okay, no No organism that lacks the ability to carry out photosynthesis has this enzyme. This is unique to photosynthetic organisms. All right, so that's step one. We got to attach carbon dioxide to a 5-carbon molecule. The next step is that basically that falls apart. It's very unstable. Once you attach that 6th carbon to that molecule, it immediately falls apart, producing 3 molecules of phosphoglyceraldehyde, which then gets reduced into a G3P, glyceraldehyde 3-phosphate. Where else have we seen these molecules? G3P and PGA. What's that?
Substrate level phosphate.
Yeah, in glycolysis, right? And so those 2 molecules are also present in glycolysis. And so this allows the actual plant to make a choice of whether to keep going through photosynthesis or to shuttle these molecules into glycolysis, knowing that plants carry out both. So that's step 2. Basically, the 6-carbon molecule falls apart and you reduce the carbon to produce G3P. And then step 3 is we need to regenerate RUBP. And so an additional reaction is going to regenerate RUBP from 5 molecules of G3P. It's going to generate 3 molecules of RUBP. Yeah.
So Rubisco isn't found in chemosynthetic organisms?
I don't believe so. Yeah, I don't believe so. All right. So this is something to keep in mind, this reaction in this statement. It requires 6 turns of the Calvin cycle to produce 1 6-carbon sugar. 6 turns of the Calvin cycle to make 1 6-carbon sugar. So that means every 1 turn is generating what? 1 carbon sugar. Except for it doesn't actually work that way because you have to be able to release it as a certain molecule, and that's G3P. So you actually have to carry out the Calvin cycle 3 times to release 1 G3P. And so we talk about that photosynthesis produces glucose. You may not talk about that, but others talk about that photosynthesis produces glucose. It's actually not true. Photosynthesis produces G3P, and another reaction will take 2 molecules of G3P and make glucose. But it's not actually part of photosynthesis. So this is what happens for every 1 turn of the Calvin cycle. You take 1 molecule of carbon dioxide, you take 2 molecules of NADPH, really just the electrons and the hydrogen from them, 3 ATP to make 1 carbon sugar, 2 molecules of NADP plus that can be reused in the light dependent reactions, and then 3 ADP. And so here is that in picture form. And so here's step 1, where you fix the carbon. Step 2, where you reduce the carbon. And then step 3, where you regenerate RUBP. And you've got to carry this process out 3 times to get 1 G3P to leave. And so you've got to carry it out 6 times to generate enough G3P to make 1 6-carbon sugar. Okay? So 3 trips around this circle release 1 G3P, and the other 2 G3P will go into regenerating RUBP. All right. No, the other 5, because 3 turns around generates 6 G3P. 5 go into remaking RUBP. 1 leaves the cycle and can be used to make glucose. Okay. Any questions? I know it's a lot, but when you sit down and you look at it, when you're not so overwhelmed with thoughts of what you're going to be doing in a few days and thoughts of all you have to do to try to get caught up while also enjoying spring break, it's manageable. It's manageable. And I know that because I was not a very good student and I managed it. Okay? I managed it. All right, so how does the Calvin cycle produce sugars and other molecules? This is just kind of summing up the thoughts, and then we'll talk about, okay, why do some plants have to separate these processes? So again, each turn of the Calvin cycle reduces a single molecule of carbon dioxide. Each turn requires 3 ATP and 2 NADPH. So 3 turns reduce 3 molecules of carbon dioxide, requiring 9 ATP, 6 molecules of NADPH. Every 3 turns, again, 1 molecule of G3P leaves, the other 5 go into regenerating 3 molecules of RUBP. And then G3P can be used to make monosaccharides. The plants can use it to make amino acids. The plants can use it to make lipids. The plants can use it to make nucleic acids. Any of those 3. All right. So if 3 turns of the Calvin cycle require 9 ATP, how many ATP are required to make 1 molecule of glucose? Yeah, if 3 turns of the Calvin cycle required 9 ATP, how many ATP are required to make 1 molecule of glucose? Or the equivalent, 18, right? Because you need 2 molecules of G3P to make 1 glucose molecule, right? If it requires 9 to make 1, it requires 18 to make 2, which is the equivalent of glucose. How many ATP are released during cellular respiration if you carry out all of the processes? Which plants are always going to carry out their respiration in the presence of oxygen because they make oxygen by splitting water, pulling electrons away from water. So plants are really never hurting for oxygen. So plants are always going to carry out respiration in the presence of oxygen. How many ATP do we make?
36.
Anywhere from 32 to 38, depending on the conditions. So which means the plant requires 18 to make it, but generates at a minimum 32. That's good. How is that possible? Because light's providing the difference. So they're releasing more than they required to make it because light provided the difference. Plus light provided the energy to make the ATP in the first place. That's good. That's good for plants. It's also good for us. Knowing that plants use about 90% of the sugars that they make for their own uses, leaving the other 10% available for the things we eat. Or for us. All right? Here's some more diagrams. It's basically the same thing. Okay? Are there any questions about the light-independent reactions? So if you were asked, I don't know, a question on a hypothetical exam that may or may not come 2 weeks from Monday, to give the 3 steps of the light-independent reactions, how would you answer that? What are the 3 steps? Step 1, you fix carbon dioxide, right? Attach carbon dioxide to another molecule. Step 2, reduce the carbon. Okay, and then step 3, regenerate what you need to do step 1 over again. Because you've got to do step 1 3 times just to get 1 molecule of G3P. You've got to do step 1 6 times to get the equivalent of glucose. All right? Does that make sense? No? Yes? A little bit? You're like, your mind's already off somewhere else, right? On the road driving to wherever you're going, or on an airplane that you're going to take, 15 minutes from now. You're really going to chance it. I one time showed up to an airport 10 minutes before my plane took off. Yeah. It was awesome. We made it on the plane, but it was, I don't know if you guys know this reference, but you should because it's a very popular Christmas movie. We had to home alone it all the way to the gate. It was awesome. It was awesome running it through, but we made it by the skin of our teeth. So, all right. I know your mind's elsewhere, but we've got a couple more questions to deal with, and then we'll be done. So what is the C4 pathway, and why is it necessary in some plants? So remember, rubisco is unique to photosynthetic organisms, and it's the site of regulating this process. It's activated by the presence of NADPH and ATP. So if you're not carrying out the light-dependent processes, you're not going to be able to carry out the Calvin cycle. And so what you'll see when you look at plants is plants that don't have a way of storing up enormous amounts of carbon dioxide. At night, they actually run out of the ability to keep carrying out the Calvin cycle because they run out of NADPH and they run out of ATP. So this enzyme Rubisco, it can fix carbon dioxide by attaching it to RUBP, but it actually has a higher affinity for oxygen and would selectively fix oxygen rather than carbon dioxide. And when it does this, is called photorespiration rather than the light-dependent processes of photosynthesis. Now, in most plant species, there's always way more carbon dioxide than there is oxygen. Because as the plant is generating oxygen, that oxygen is diffusing out of the tissues really fast. Because there's very little oxygen in the surrounding air because that oxygen is being used up by the things eating the plants. Okay, so most plants, you have way more carbon dioxide than you do oxygen, making it very unlikely that photorespiration is going to happen because there's just not very much oxygen present in the tissues. But plants that live in really dry areas that are drought tolerant actually have to keep their stomata closed during the day to prevent water loss. Very little water in the soil, so there's very little water in the plant tissues. If their stomata are open, they're going to lose what little water that they have. Okay? And if their stomata are closed, oxygen is not diffusing out, carbon dioxide is not diffusing in, meaning these plants build up an enormous amount of oxygen inside their tissues, making photorespiration extremely likely and going to outnumber the light-independent portions of photosynthesis. So plants that have this condition that keep their stomata closed during the day, they have what's called the C4 pathway. So in the C4 pathway, it gives you a way of trapping carbon dioxide and storing it up for later use. So say at night when your stomata are open, you can fix and attach a whole bunch of carbon dioxide and trap it inside of the plant's tissues for using the next day. And so this fixes carbon dioxide much better than the Calvin cycle does. It does require more energy, which is why it's costly to be drought tolerant, and most plants, if they don't have to be, will not be, because it's energetically costly. And so during the C4 cycle, instead of carbon being attached to RUBP, which immediately breaks down into two molecules of 3-PGA, carbon is fixed onto oxaloacetate, and then which immediately gets reduced by adding electrons to it into malate. By the way, these molecules are part of the Krebs cycle, and so plants have these readily available, and then it can be stored as malate. And then when it's ready, malate can be oxidized by having the electrons stripped away, generating pyruvate, which can also be used, or it can be oxidized further to generate the Krebs cycle, and then releasing carbon dioxide. So this gives the plants a way of storing up enormous amounts of carbon dioxide for later use. And again, it's energetically costly, but sometimes necessary if the plants have to keep their stomata closed during the day. And so here's the process of fixing carbon by Rubisco. Here you have RUBP and carbon dioxide generating 2,3-phosphoglyceraldehyde or glycerate is a conjugate base. And then so here is the fixation of oxygen by Rubisco rather than the fixation of carbon dioxide. All right. So the last question we're going to deal with is where and when does the Calvin cycle have to happen? So the Calvin cycle, sometimes called the C3 cycle, happens in all plants. The question is, does a plant need to have an additional cycle to trap carbon? But the only way to actually generate the G3P that you need to make the equivalent of sugars, you have to have the C3 pathway, or the Calvin cycle. So in most plant species, C3 plants and CAM plants both the light independent and the light dependent reactions happen in all photosynthetic cells. I don't know why that says plant dependent reactions. That's awesome. In C4 plants, the Calvin cycle happens only in really deep cells. The Calvin cycle happens only in really deep cells, whereas the C4 pathway and the light-dependent reactions happen in all photosynthetic cells. In C3 and C4 plants, carbon fixation and the Calvin cycle happen during the day, which leaves us with CAM plants, where something has to happen at night and not during this day. So in CAM plants, CAM stands for crassulacean acid metabolism. They fix carbon at night when the stomata are open and then store up that carbon dioxide in the central vacuole of the plant. So then during the day, the malate can come out of the central vacuole, be reduced into pyruvate, releasing carbon dioxide. Okay. The remainder of the slides on here just show you some of the different anatomy and some of the different mechanisms by which these different plants function. But I'm actually going to close and end our class with this. I just want you to know that for plants that are drought tolerant and have to keep their stomata closed during the day, they have to have some mechanism by which you can separate carbon dioxide from oxygen. Okay, some plants, their strategy is to bury carbon dioxide really deep in the tissues where you're not getting very much of the light dependent reactions happening. And so if the light dependent reactions aren't happening, you're not generating a lot of oxygen. And carbon dioxide is buried, sequestered down deep in the tissues where you're not generating a lot of oxygen. Other plants, their strategy is to trap a bunch of carbon dioxide at night, release it during the day, just overwhelming the amount of oxygen that's present. In both those strategies, the stomata are closed during the day to prevent water loss. All right, so oxygen is not diffusing out, carbon dioxide is not diffusing in. But it's two different strategies to ensure that where rubisco is, there's only carbon dioxide, or way more carbon dioxide than there is oxygen. All right? Any questions, thoughts, concerns, things you want to share with the group? All right, well, we're going to end here. We're about 6 minutes before our scheduled end time. If you want those 6 minutes, stick around. I will give them to you. If you don't, enjoy your spring break. Yeah. Yeah, so the two strategies are, one, bury all the carbon dioxide really deep in the tissues where very little of the light-dependent reactions are happening. Or trap so much carbon dioxide at night that it can be released during the day, just overwhelming the amount of oxygen that's present.
Are we meeting today still?
Yes.
Okay.